flow or the path of circulation of cerebrospinal fluid. So I'm going to walk you through this so you can see where it's made, how it flows through those ventricles, and then how it actually gets out of inside the brain ventricles and actually around the brain and spinal cord. Actually, I think I have a little video here. All right. So if you watch this, I'll turn the light off. It's really short, but it'll kind of show you where the cerebrospinal fluid is made and then how it circulates inside the brain ventricle system, and then it leaves the brain and then actually goes outside and surrounds the brain and spinal cord. So we'll watch that a couple times. Oh, dark. Wow. And then we'll go through and talk about that. There's no sound. One more time. So we can see several things on this, and I'll point them out on the other diagram. Here's your arachnoid granulations. Here's your superior sagittal sinus, right here. These structures right here in the lateral ventricle are kind of red and blue, and down here in the fourth ventricle, those are the structures that make the cerebrospinal fluid. And then what we see, there's specific locations where that cerebrospinal fluid actually can leave that inner ventricular system of the brain and actually flow around the surface of the brain in the subarachnoid space. All right, let's go ahead and watch your eyes. All right, so the cerebrospinal fluid is made by structures called the choroid plexus. And essentially these choroid plexi, or plexuses, are blood capillaries surrounded by ependymal cells. Remember those? Yes. One of the 6 types of neuroglial cells. These specifically are central nervous system glial cells. So surrounded by ependymal cells. So there's 3 places where we find these choroid plexuses. In the lateral ventricles, which are the hollow spaces inside the cerebrum. We find them in the third ventricle. And really what it is, it's just kind of an extension of the ones in the lateral ventricle. And we also find the choroid plexuses in the fourth ventricle. So there's three different places where the cerebrospinal fluid is made. Now keep in mind, there's a fixed volume or fixed amount of empty space that can be filled with fluid. And these things are constantly producing cerebrospinal fluid. They do it by filtration. The blood flows through these capillaries, and the pressure forces, really, blood plasma out of the blood, not the blood cells, but the blood plasma out of those capillaries, through those ependymal cells, and then into those spaces. So cerebrospinal fluid is very similar in composition to blood plasma, and you can actually diagnose a fair amount of things by analyzing that cerebrospinal fluid. Of course, we know that you can get that cerebrospinal fluid by aspirating the fluid from the subarachnoid space with a needle, typically down there in between L4 and L5, because there's no spinal cord there to damage. Yeah.
You said it constantly produces cerebrospinal fluid by diffusion?
Filtration. Filtration. I'm going to put numbers on these. I don't know how many I'm going to end up with, but so we'll just say that we're going to start up here with the choroid plexus in the lateral ventricles. So we'll say CSF is produced by CP, by the choroid plexuses in the lateral ventricles. Now that cerebrospinal fluid leaves the lateral ventricles via an opening called the foramen of Monroe.
In the book they call it interventricular foramina.
The interventricular foramina. This is what we call an eponym, named after somebody. Same thing. So it leads via the interventricular foramina, or the foramen of Monroe, to the third ventricle. Now what else is going to happen here, based on what you see in the diagram? Cerebrospinal fluid is made in the lateral ventricles, leaves via the foramen of Monroe, enters the third ventricle, and we're going to get additional cerebrospinal fluid added here at this point. So additional cerebrospinal fluid added in, or by, in is better, I guess, sorry, in the third ventricle. Why? Because there's another series of choroid plexuses there. Because there's another set of choroid plexuses in the third ventricle, the structures that make that cerebrospinal fluid. All right, so where would it go from here? From the third ventricle, that fluid would leave and enter this little channel here.
Central canal.
Not the central canal.
Fourth ventricle.
Not yet. It's got to get to the fourth ventricle first.
Okay.
Cerebral aqueduct.
See, it doesn't say that. Oh, yes, it does. 4, yeah.
Enters the cerebral aqueduct, which takes it to the fourth ventricle. Now this is the point where we start to see that cerebrospinal fluid leaving inside the brain and starting to diffuse out or be forced out into that subarachnoid space. I think I'm number 5. So at this point, some of the CSF will...
Is there more antidote?
Yes, I'm not quite there yet, but yes. Yeah, there is. So at this point, some of the CSF will enter the subarachnoid space. Now there's a little opening, or 2 little openings, on the lateral sides of that cerebral aqueduct, just at the point where the cerebral aqueduct becomes the 4th ventricle. And those are called lateral apertures. Little openings. So as you're studying this and learning this, miniaturize yourself, like the 1987 movie Interspace with Martin Short. He gets miniaturized in this little tiny capsule, and he's injected into somebody's blood, and he's traveling through this person's body, and he has to get out before the miniaturization wears off. Okay? So miniaturize yourself and pretend you're in a little capsule, and what are your options as you're traveling through this ventricular system? Or don't. So that's how it gets into, one of the ways it gets into the subarachnoid space is through the lateral apertures. Okay, so in the 4th ventricle then, more CSF is added. Why? Because what do we have here? Another set of choroid plexuses, or another choroid plexus. Now from here there's 2 options. From the 4th ventricle there's 2 options. 7A. CSF leaves via what's called the median aperture and enters the subarachnoid space. So this one's kind of right in the middle. Kind of inferior to the cerebellum there. Leaves via the median aperture into the subarachnoid space. That's 7A. 7B. CSF continues down into the central canal, the spinal cord. Now once it's in that subarachnoid space, it will circulate through the subarachnoid space, and this can be number 8. So the CSF circulates through the subarachnoid space.
part down spinal column does it circulate?
It will circulate all the way down into the sacrum where those spinal nerves are. They're all going to be bathed in that. Or now, wherever there is dura and subarachnoid space, it's going to go. So, pretty much all the way down.
So it all returns basis.
Right. Yep. Yep. It goes down and then it's forced back up and out. Yep. So it looks like it comes down one side and goes around the spinal cord and then gets sucked back up.
Does it have a lifespan?
Well, however long it takes to go from here to here. Could be a matter of minutes. So 9 then would be that cerebrospinal fluid is then absorbed or forced through the arachnoid granulations into the superior sagittal sinus.
One more time.
The cerebrospinal fluid is then absorbed or forced through those arachnoid granulations into the superior sagittal sinus. Remember those arachnoid granulations I mentioned way at the beginning?
You're calling them in the book dural venous sinuses.
Same thing as that's the superior sagittal sinus is one of the dural venous sinuses.
Okay.
That's the one where cerebrospinal fluid enters.
Thank you.
Here is an arachnoid granulation. The arachnoid mater pops up through the dura into that superior sagittal sinus, and that cerebrospinal fluid is forced between the cells that make up the arachnoid mater, and then that fluid can then circulate and travel within that venous blood, where any wastes that are in that cerebrospinal fluid are processed as the blood flows through the rest of the body. Well, to the heart, and then to the lungs, and then to the rest of the body.
Okay, that's where the wastes are picked up then.
Picked up. Cerebrospinal fluid, which picks up the waste from the various cells of the brain and spinal cord, and as the cerebrospinal fluid enters the superior sagittal sinus, the blood is going to take that cerebrospinal fluid throughout the body, and various cells in the body, probably in the liver and other places, are going to take out any waste products that might be in that.
Thank you.
So it's really important that there's kind of a constant pressure that's maintained, a cerebral spinal pressure. And that pressure can be measured. I don't know what the standard pressure is. It's not important for our purposes here. But we have to balance the amount of fluid production with the amount of fluid that's lost. If there's more fluid that's produced than what's lost or reabsorbed, then you got too much fluid and that builds up the pressure. If you have not enough fluid production, then you're not going to have enough buoyancy for the brain and the spinal cord, and they're going to begin to settle. So we have to have a nice balance between production and loss.
It's funny you mentioned that. I just wanted to refer this young man when I was working nights patient transport. He was producing too much cerebrospinal fluid, and they had to put in 3 shunts in a matter of 5 years from the brain to the stomach because the fluid kept building up, and the brain surgeon would go ahead and reestablish the shunt into the stomach so it could reabsorb the cerebrospinal fluid. How that was done, I don't know. But
there are conditions where people do produce too much cerebrospinal fluid, or there's conditions that result because cerebrospinal fluid is not circulating properly. I know some people with spina bifida, their cerebrospinal fluid doesn't circulate properly. And so in order to prevent the increased pressure in the brain and destroying those brain cells, they put in a shunt that will take some of that excess cerebrospinal fluid and drain it typically down into the digestive system or somewhere in the abdomen where it's then absorbed by the cells. So that's what they did. So the shunt would then allow more fluid to be pulled out of circulation to match what is being produced.
How would you know that it's producing too much?
Um,
Is there like signs?
I'm sure there are. hydrocephalus is what the condition is called. I don't know what the signs and symptoms are. I think when a baby's born, I think they can kind of tell. One of the things that happens in a baby, you have the fontanelles, the soft spots, okay? Those will kind of start to bubble, and that's kind of a sign. Yeah, because there's just connective tissue there. And so as that fluid builds up, the brain will actually kind of be forced and pushed up through that. That's one sign. I don't know what it would be in adults. Probably a lot of headaches because of the pressure that's built up.
His balance was off, too, a lot, this young man, until he got the shunt in.
So this is a picture that shows you what happens to the brain of someone with hydrocephalus. Okay, hydro meaning water, obviously. So here's the normal brain ventricles. Notice that the ventricles in the hydrocephalus person are quite swollen and enlarged, and that can actually force then those lateral ventricles and push the walls of those and damage those brain cells associated with those ventricles and around those brain cells. So they shunt it. They create a shunt to get rid of that excess fluid to help maintain that pressure. I would say yeah, but like Pat says, and they sometimes may have to go in and be replaced, but yeah, in order to, if a person has excess fluid because of perhaps spina bifida or something where, the body, the cells don't know that, they're producing the amount that they're supposed to, but with spina bifida, there may not, the volume, the space in that subarachnoid space is smaller, so the fluid can't, there's too much fluid to fit. So yeah, there's going to have to be something done to constantly maintain the proper pressure. All right, I want you to go ahead and just kind of review this path of circulation. Think about the different anatomical structures that this fluid is going to be associated with. The ventricle system, the choroid plexi. Notice on the diagram the different brain structures that are going to be bathed by this cerebral spinal fluid and its relationship then to the superior sagittal sinus here. Okay? We'll take a couple minutes and do that.
So that's superior sagittal sinus. They say that's why you get headaches when you're drunk. See this stuff? No.
Oh, really? Maybe it's a chemical inhibitor with the choroid plexus that limits the amount of production of the cerebral spinal fluid initiated by the alcohol. Does it? Well, as far as he said, sometimes your cerebrospinal fluid will become, you won't produce as much when you're drinking alcohol. Is that because of the choroid plexus, because of the chemical stimulant wouldn't be producing enough cerebrospinal fluid?
Got me.
The choroid plexus, it actually produces the CSF.
Via filtration. Yeah, it's
Via
like
filtration.
yeah, it's just like the capillaries in your tissues. And how your tissues get the substances they need. Things are forced out of the blood via filtration. The blood cells and the large proteins are kept in the blood. The other small things are filtered out.
See, it's
Same
just showing
idea.
a picture here of the choroid plexus, but it doesn't. I'd love to see it how it actually produces and puts the fluid into the ventricles.
It's producing it just like I said. It's just being the hydrostatic pressure of the blood is forcing those substances out through those capillaries,
Okay.
and it's basically blood plasma.
Okay, so the CSF actually is blood plasma.
Yeah, it's very similar.
Right. Just to clear it. Okay.
There may be some differences in the protein consistency that's in it, but maybe certain things are not filtered out in the choroid plexuses that are filtered out in other blood capillaries.
So it's actually set up the choroid plexuses are like filters for the blood to bring it into the.
Just like in the kidneys, just like in every other
Okay.
capillary bed in the body.
very, very.
Any questions about this fluid flow process?
One question.
Yeah.
It only returns to the interior portion from the spinal cord. Is that what it looks like? It has a choice of different spots where it can look to the back.
Well, keep in mind that the spinal cord, you have this subarachnoid space that's completely around it. So as that fluid is leaving through that median aperture, okay, it enters that subarachnoid space. So the spinal cord is kind of floating. There's like a space around it. So that fluid can flow down and then flow around and then flow back up as well.
Does it technically have to go all the way down before it comes up or no?
Probably not. But there's going to be... Cheyenne, Corky, you here? Yes? You need to get out your stuff. Is she here? Cheyenne, Corky? She's not here? Yeah, that's all right. Okay. What were you saying?
So then it would return only out to somewhere.
Correct. Yeah.
There's no
Circulation.
There's no little cranial.
No, that's where they are, up there.
There's none lower. They're all up there.
I mean, there may be some small ones out here,
But they're all.
but
There's none in the spinal column. So.
no, no. No, and to answer your question, Nina, obviously there's going to be enough, it's going to be forced down to the bottom. There's like a current. Remember, the ependymal cells, which line these cavities, have little cilia on them. And so they're creating this current that forces this fluid out and keeps it flowing and moving.
Oh, one more question. Can you explain the diffusion aspect?
Diffusion of what?
The cerebrospinal fluid, how it diffuses in? Pardon me.
Diffuses from where? It's produced by filtration, and then
Filtration, okay.
this is, in a way, it's not. This would be a diffusion process here, but it's just being forced via pressure, and that fluid's being forced out into the blood, into that.
Okay.
Yeah. So.
At the arachnoid granulation.
Right.
You mentioned it.
Right. Yeah.
Thank you.
Yeah. So yeah, I mean, it's diffusing from this place to this place, but not necessarily because it's higher concentration. In this case, diffusion probably wasn't the right word. It's being forced